Today's video is a discussion on gluconeogenesis. Gluconeogenesis is the metabolic pathway for the synthesis of new glucose molecules from non-carbohydrate precursors. Non-carbohydrate precursors can be lactate, glycerol, or pyruvate, as we are going to see. Gluconeogenesis is the de novo synthesis of glucose, meaning that it is not the glucose molecules are not produced by the breakdown of glycogen, which is present in our skeletal muscle cells or liver cells. Instead, non-carbohydrate precursors which contain carbon are utilized to make new molecules of glucose. Gluconeogenesis works to elevate the levels of blood glucose, and so in our cases particularly, it is functional whenever there is condition like starving or intense exercising or a low carbohydrate diet or even fasting. So in these particular situations, our blood glucose level tends to go down, and after some time when our glycogen stores have been exhausted, the body makes new molecules of glucose via gluconeogenesis process. Gluconeogenesis primarily occurs in liver cells, and to some extent it is also seen in kidney cells and small intestinal enterocytes. The following molecules serve as precursors for gluconeogenesis. Pyruvate is formed from the oxidation of glucose via glycolysis. Lactate is a product of lactic acid fermentation, and it is actually formed from pyruvate itself. Glycerol is formed from the breakdown of triacylglycerides which are present in the adipose tissue. Glucogenic amino acids are those amino acids which can be utilized to form glucose. So they make one or the other product of TCA cycle or a pyruvate molecule, and ultimately they can be utilized to form glucose. There are two amino acids, however, which are exclusively ketogenic, and so they cannot form glucose, and those are lysine, so that's a K, and leucine, which is an L. These are the single letter abbreviations. And finally, we have propionate. Propionate is a small chain fatty acid, and it is formed as a product of the oxidation of fatty acids containing odd number of carbon atoms. Propionate serves as a major precursor for gluconeogenesis in organisms like ruminants. In our case, it serves as a minor precursor for gluconeogenesis, not a major one. Before we discuss the various substrates, let's just look at the overall process of gluconeogenesis. Now, I'm assuming that all of you must know glycolysis at this point. So in this particular diagram, we can see how glycolysis and gluconeogenesis are kind of opposite processes to each other. So if you go this way, you are actually making two pyruvate molecules from the oxidation of one glucose molecule, and that is glycolysis. But when you go this way, you are utilizing two pyruvate molecules to make one molecule of glucose, and that is gluconeogenesis. But strictly speaking, these two processes are not thermodynamically reversible of each other. Why is so? Let's look at this. In glycolysis, if you recall, there are three steps which are highly exergonic. Where is my figure? Okay. So in glycolysis, if you recall, there are three steps which are highly exergonic, and these three steps, because they have very negative delta G's, they are irreversible. So the step number 1, that is the phosphorylation of glucose. Notice how its delta G is very negative. Similarly, you have step number 3, in which you are phosphorylating a fructose 6-phosphate molecule. That is also an exergonic reaction. And similarly, if you talk about the last step of glycolysis, in which you are converting a PEP molecule into pyruvate, that process also tends to release energy. So these three reactions in glycolysis are irreversible, and so in order to form a glucose molecule, you need to bypass these reactions. Okay, we need to devise other ways or other reactions by which we can overtake these reactions in order to produce a glucose molecule. For all other reactions of glycolysis which are shown in black, you can see that either they are endothermic, or actually the better word is endergonic, meaning that they have positive delta G's, or even if they have a negative delta G, but that's not a very high negative delta G, so you can overtake them with the help of the same enzyme. So these reactions can go backwards, right? So there is nothing as overtaking them. These reactions can go essentially backward because they look at this molecule. This can even have a delta G of 0, so tends to exist at an equilibrium in physiological conditions. So all of these reactions you can go backwards, but in all the red reactions, because they are highly hexagonic, you cannot essentially go backwards. And so we have to devise some reactions which are known as the bypass reactions. Coming back to this diagram, let me change the ink. Let's look at the bypass reactions. The first bypass reaction is when you are trying to convert pyruvate into phosphoenol pyruvate. And this reaction is itself composed of two sub-reactions. The first one is when you are converting pyruvate into oxaloacetate. And the second reaction is the conversion of oxaloacetate into phosphoenol pyruvate. And notice how you have an oxaloacetate molecule as an intermediate of gluconeogenesis. Oxaloacetate is also an intermediate of TCA cycle. So TCA can also be, a source of oxaloacetate. Or if I say it in other words, this reaction is an anaplerotic reaction which can be used to generate oxaloacetate molecules which can be fed into the TCA cycle, right? So it can be used as an anaplerotic reaction. The enzyme which catalyzes this reaction is pyruvate carboxylase, and pyruvate carboxylase, as its name suggests, is going to carboxylate a pyruvate molecule. Pyruvate carboxylase, which we call PC, is actually called an ABC enzyme. It's called an ABC enzyme. And the reason why it's called an ABC enzyme is A stands for ATP, B stands for biotin, and C stands for carbon dioxide. So as you can see over here, the pyruvate is being converted into oxaloacetate, and this is an endergonic reaction itself. The energy for this reaction comes from the hydrolysis of an ATP molecule. So you can see two molecules of ATP because you have two molecules of pyruvate converted into two molecules of oxaloacetate. So per pyruvate molecule, you are going to require an ATP molecule to convert it into oxaloacetate. Apart from that, since pyruvate carboxylase is a carboxylase, like all other carboxylase, it also has a prosthetic group which is biotin, and biotin is the one which is actually going to bind the carbon dioxide, which is then transferred onto the pyruvate molecule. The C stands for, as I just said, carbon dioxide, and carbon dioxide is actually present in the form of a bicarbonate ion. So it's really the bicarbonate ion which you are utilizing, and that's not shown in this diagram. The pyruvate carboxylase is going to carboxylate pyruvate, and the source of carbon dioxide is going to be a bicarbonate ion. Next reaction, you can say the next sub-reaction, we are converting this oxaloacetate molecule to phosphoenol pyruvate. This reaction is also an endergonic reaction, and in this process you utilize a GTP molecule, you hydrolyze the GTP molecule, as well as you transfer a phosphate from this GTP molecule on a pyruvate. So you get phosphoenol pyruvate as a result. And this reaction is catalyzed by PEP carboxykinase, which is known as PEP-CK. And PEP-CK is also an interesting enzyme. It's interesting because it exists in two isoforms. You have an isoform of PEP-CK present in the mitochondrial matrix of hepatocytes, and you have an isoform of PEP-CK present in the cytosol of hepatocytes and other cells where gluconeogenesis is taking place, obviously. So this reaction can thus take place either in the mitochondrial matrix or in the cytosol, and that is special, and we are going to see why that is special in just a moment. But for now, this step should be clear that you are taking GTP, you are using it as a phosphate donor, and you are using PEP carboxykinase to catalyze this reaction. And now we have essentially bypassed the first reaction. We have converted pyruvate into phosphoenol pyruvate. Another thing that I would like to mention over here is the location of the enzyme pyruvate carboxylase. Pyruvate carboxylase is present in the mitochondrial matrix only, and thus this reaction, the conversion of pyruvate to oxaloacetate, can take place only in the mitochondrial matrix. This reaction, however, the one catalyzed by PEP carboxykinase, as I just said, it can take place in mitochondrial matrix or it can take place in the cytosol.
Sorry guys, somehow my video got cut, so everything that I wrote on the screen is somehow gone. But as we were discussing that the pyruvate carboxylase is present in the mitochondrial matrix, and so this reaction can only take place in the mitochondrial matrix, whereas PEP carboxykinase is also present in the matrix as well as it is present in the cytosol. So this reaction can take place in the cytosol as well. And this is important. We can see that gluconeogenesis is a reaction which is taking place in different areas within the cell. This is unlike glycolysis, which is, by the way, we go from glucose to pyruvate. In glycolysis, all these reactions were taking place in the cytosol itself. And all of these, so glycolysis is essentially a cytosolic metabolic pathway. It is occurring in cytosol entirely, but gluconeogenesis is a compartmentalized reaction. Some part of it is taking place in the mitochondrial matrix, some part of it is taking place in cytosol, and as we are going to see, the last step, which is this step, right, the third bypass reaction, this step takes place in the ER, the endoplasmic reticulum. We are going to discuss it later, but for now, let's come back to the fact that this gluconeogenesis is taking place in different compartments within the cell, and that is actually an important thing because that helps us to regulate glycolysis and gluconeogenesis, because we want to avoid glycolysis and gluconeogenesis occurring at the same time within the cell. If they're occurring at the same time, then there is no net production of ATP and there is no net production of pyruvate. So actually gluconeogenesis utilizes more ATP than is generated by glycolysis, and so if these two reactions are occurring at the same time, that can be very energetically unfavorable and that can be energetically expensive process for the cell, and that is why we need to avoid these two reactions occurring or taking place in the same area within the cell. So this has been discussed already, that the PEP carboxykinase reaction. Now the PEP carboxykinase does its job. You get a phosphoenol pyruvate, and these reactions can actually, all of these reactions can take place, and they take place in the cytosol. So they take place in the cytosol. Now here's the thing. When you are converting PEP carboxykinase to phosphoenol pyruvate within the mitochondrial matrix, it makes sense. Phosphoenol pyruvate is going to leave the mitochondria and it's going to enter the cytoplasm or the cytosol, and all of these reactions are going to take place. But what if it's the oxaloacetate which is leaving the mitochondria? If you are converting oxaloacetate into phosphoenol pyruvate inside the cytosol, in that particular case, you're going to use the PEP-CK C isoform, that is the cytosolic isoform. In that particular reaction, there is one major difficulty. Oxaloacetate cannot leave mitochondrial matrix. The membrane of the mitochondria does not have any transporter molecule for oxaloacetate transport, nor is oxaloacetate a molecule which can freely diffuse out of the mitochondrial matrix. There has to be a way by which you can transport oxaloacetate outside, and that actually occurs in a separate reaction, and that is the conversion of oxaloacetate into a molecule which is known as malate. And recall that this reaction is actually also, this is a reaction which you must have studied in while studying the citric acid cycle. This is a reversible reaction, and the reaction is catalyzed by the enzyme malate dehydrogenase. For this particular reaction for making malate from oxaloacetate, we are going to utilize two NADH molecules which are going to get oxidized into NAD plus molecules. So over here, you're going to use two NADHs and you're going to generate two NAD plus, and obviously you're going to have two oxaloacetate, so two malate molecules. And it is the malate which is going to leave the mitochondrial matrix, because malate, there is a transporter molecule in the membrane of mitochondria which can transport malate outside into the cytosol. So malate is actually able to leave, and once within the cytosol, this malate is going to get converted back into oxaloacetate, because malate dehydrogenase, the enzyme catalyzing this reaction, is also present in the cytosol. And once this reaction occurs, you are essentially going backward. So you're going to use two cytosolic NAD plus, right? So let's just write this way. So you're going to use two cytosolic NAD plus, and you're going to generate two NADHs. And this is important. Remember that we generate two NADH in this reaction. Where are these two NADH generated? These are generated in the cytosol. Okay, so now once we have our oxaloacetate molecule in the cytosol, it can get converted into phosphoenol pyruvate, right? Essentially, we now know that the first reaction has been bypassed. We have formed phosphoenol pyruvate, and we have formed phosphoenol pyruvate from pyruvate molecule. All of these reactions are occurring in the cytosol. Our phosphoenol pyruvate molecule is in the cytosol, so no worries, these reactions can proceed. However, notice this reaction, right? In this reaction, you see how you have to feed two ATP molecules, because you have to generate a 1,3-BPG molecule from 3-PG molecule. And so you utilize two ATP molecules over here. Also, in this reaction, when you go from 1,3-BPG to glyceraldehyde 3-phosphate, you use two NADH molecules, right? And you release two inorganic phosphate, and you also form two NAD plus molecules. Now, here is the thing. These NADH molecules over here that were formed in the cytosol are in fact the ones which are going to be fed in this reaction. Okay, so this is the essence of this reaction taking place. When you have pyruvate as your precursor, the oxaloacetate essentially does not get converted into phosphoenol pyruvate within this matrix. It leaves the mitochondria in the form of malate, and then malate converts back into oxaloacetate, so that you can generate these two NADH molecules, which can be then fed in this particular reaction. Okay, this is the essence of this reaction taking place over here. Now, once you get to 2,3-G3P molecules, one of them is going to get converted into dihydroxyacetone phosphate. It is simply going to isomerize. So now you have one GAP molecule and one DHAP molecule. It might be a little bit confusing in this diagram, but you have one GAP molecule, the glyceraldehyde 3-phosphate, and you have one DHAP molecule. Okay, and these are going to undergo aldol condensation to form this particular molecule, which is fructose 1,6-bisphosphate. These are going to undergo aldol condensation. And notice how we have, earlier we were using 2 moles, and now we have only 1 mole of fructose 1,6-bisphosphate. Why? Because you were forming 2 molecules of glyceraldehyde 3-phosphate. One of them got isomerized into DHAP molecule. So one GAP and the other GAP molecule got isomerized into DHAP. The two condensed together, and now you have a fructose 1,6 bisphosphate molecule. The next step, the conversion of fructose 1,6 bisphosphate into, let me change the ink, the conversion of fructose 1,6 bisphosphate into fructose 6 phosphate. This step is again, it's a second bypass reaction because it has had a delta G of a negative value. So this step is catalyzed by an enzyme, fructose 1,6 bisphosphatase 1. And unlike in glycolysis, you were using an ATP molecule and you were generating an ADP molecule. But in this process, you're not going to generate ATP molecule. It's a simple hydrolysis reaction. You use a water molecule, you release the inorganic phosphate, and you get the fructose 6 phosphate molecule. The next reaction is a simple isomerization reaction. You get glucose 6 phosphate. And the third bypass reaction, this particular step, it's a very interesting step. And this enzyme, glucose 6 phosphatase, is also a very interesting enzyme because it's an enzyme which is present as a part of a protein complex which is present in the ER membrane. So this reaction can only take place in the endoplasmic reticulum of hepatocytes and kidney cells where the gluconeogenesis is taking place. This is very important. Why are we making this reaction? Why is this reaction occurring in the ER? Why is this reaction not occurring in the cytosol like the other reactions? The reason is, recall why were we making glucose 6 phosphate molecule in the very first place. We were phosphorylating the glucose molecule for two reasons. Number one, we wanted to trap that glucose molecule inside the cell so that it could not leave the cell. Addition of a phosphate group was making it more polar, and so it was trapping the molecule inside the cell. Polar molecules would not be able to escape lipid bilayer easily. The other reason why we were doing so was addition of phosphate group was making the molecule even more reactive, and so the glucose 6 phosphate could be broken down into pyruvate. You could generate energy, and that energy could be used to drive all the reactions of the cell. So that is why this reaction, this step of phosphorylation of glucose was taking place in the very first place. Now, if this enzyme is present in the cytosol, what would be the result? You would form glucose back from glucose 6 phosphate. It would leave the cell, and the purpose of phosphorylation would actually fail. It would not make sense. Why would you phosphorylate it? So to prevent that, this particular enzyme and this particular reaction take place only in the hepatocyte's endoplasmic reticulum. And what it does is that because hepatocytes and the kidney cortex cells are the ones which are going to regulate the blood glucose levels, whenever the blood glucose levels go down, whenever you are facing a hypoglycemic condition, these cells are going to sense it, and then this reaction is going to take place in the mitochondrial matrix. The glucose 6 phosphate molecule, it's going to enter the ER. Did I say mitochondrial matrix before? I would like to correct myself. The glucose 6 phosphate molecule is going to enter the endoplasmic reticulum, and once it is inside the endoplasmic reticulum, it is going to undergo dephosphorylation. However, this is a simple hydrolysis reaction once again, and so you use a water molecule and you release a phosphate, an inorganic phosphate, to ultimately form a glucose molecule. So you can see how compartmentalized gluconeogenesis process is. Just a quick revision, it is taking place in 3 locations. The first reaction, pyruvate carboxylase, takes place in the mitochondrial matrix. The second reaction from here to here, it can take place in the cytosol, and this final reaction takes place in the ER. So it's compartmentalized, unlike glycolysis, which entirely takes place in cytosol. Now this is a diagram I got from the medicalbiochemistry.org page, a very good site, you can check it out. But what they have shown over here is just a detailed diagram of the gluconeogenetic chart that we just saw. You can see how the pyruvate is entering the mitochondrial matrix, right? Pyruvate was formed in the cytosol as a result of glycolysis. So pyruvate is entering the mitochondrial matrix, and you have a carrier protein, that is mitochondrial pyruvate carrier. and then this would essentially move the pyruvate inside the mitochondrial matrix. Over here, it's going to undergo the reaction. You can see how the ATP and the CO2 are being used, and you're going to form oxaloacetate, which will give you malate, because if your pyruvate is your precursor, then obviously malate has to leave. This is your carrier for malate, the transport molecule for malate. The malate goes out, gets converted back into oxaloacetate. The oxaloacetate will give you phosphoenol pyruvate. You can see the PEP-CK over here. And then all of these reactions will take place. Ultimately, you're going to have the glucose molecule. This particular reaction is going to take place in the ER. And this is the gist. Now also notice other molecules over here. Notice the glycerol molecule, the lactate molecule, and alanine molecule. Alanine is a glucogenic amino acid. And notice how all of these were also substrates. So let's now discuss what would happen if these molecules are the precursors for gluconeogenesis. Now, if lactate is a precursor for gluconeogenesis, then lactate is actually going to give you pyruvate molecule. So in our skeletal muscle cells, when we are exercising, due to anaerobic conditions, lactic acid fermentation takes place, and in that particular process, we form a lactate molecule from a pyruvate molecule. So a pyruvate molecule undergoes reduction, it gets reduced into lactate, whereas the pyruvate molecule, the NADH molecule gets oxidized to form NAD+. And these NAD+ are actually utilized in glycolysis reaction. So this particular step, as you can see, you needed NAD+ molecules, and this particular reaction, the lactic acid fermentation, happens in the very first place because the oxidative phosphorylation, the rate with which the oxidative phosphorylation is occurring, is far lesser than the rate with which glycolysis is occurring. And so to fuel the glycolysis, you need the NAD+. And to generate these NAD+, you have the lactic acid fermentation taking place. So this is the step, this is the lactic acid fermentation that takes place in your skeletal muscle cells during anaerobic conditions, and that takes place when you are exercising, you're doing intense, heavy workout. But now in this particular situation, when you use lactate as a gluconeogenic precursor, notice how this reaction is reversible. So if you can go this way, you can also go this way, and the same enzyme will be able to catalyze this reaction. The only difference is, which they have not shown, is that you're going to use an NAD+ molecule and you're going to generate an NADH. So if you go this way, this is actually going to occur like this. So you're going to generate an NADH molecule. So when your lactate is the precursor, this reaction of conversion of lactate into pyruvate will take place in the cytosol, and during this reaction you are going to generate NADH molecules. Let's come back over here. So you can see lactate was getting converted into pyruvate, and in this process you are generating NADH molecules. The pyruvate is going to enter the mitochondrial matrix, and all the steps from here are going to take place in the same way that we have discussed. The only difference over here is that this step will not occur, and eventually this step will also not occur. Oxaloacetate would form PEP while in the mitochondrial matrix. So it will form PEP inside the mitochondrial matrix, and it is the PEP which is going to leave. Now, why did we skip these reactions? The reason why we were doing these reactions in the very first place was to generate NADH molecules. And these NADH molecules were the ones which were being fed in this particular reaction. But now, since we already have NADH molecules in the cytosol from conversion of lactate into pyruvate, we no longer need these NADH molecules to be generated, and so we can skip these reactions. Instead, the oxaloacetate can be directly converted into PEP within the mitochondrial matrix, and the PEP can leave the mitochondrial matrix, and then it can undergo further reactions, because these NADH molecules, we already have a source for these NADH molecules, and that is these NADH molecules which were generated during conversion to pyruvate. And this is what is shown in this diagram. You can see when pyruvate is your precursor, then the pyruvate enters the mitochondria, it gets converted into oxaloacetate, which gets converted into malate. Malate leaves and malate gets converted back into oxaloacetate, and these NADH molecules are actually the ones which are going to feed this particular reaction in which you were converting 1,3-BPG into glyceraldehyde 3-phosphate. But if your lactate is the precursor, then it is going to give rise to NADH molecules during the conversion of lactate into pyruvate itself. So when the pyruvate molecules enters the mitochondrion, it will not undergo, or actually the oxaloacetate molecule will not undergo conversion to malate, because you already have a source for these NADHs. So it will simply get converted into PEP, and the PEP is then going to leave the mitochondrial matrix, as you already have NADH in the cytosol. Alright, so we have already discussed what would happen if lactate is the precursor, what would happen if pyruvate is the precursor. There is one important difference, and that is when your pyruvate is the precursor, it is your malate molecule which leaves the mitochondrial matrix. When your lactate is the precursor, it is your PEP molecule which leaves the mitochondrial matrix. I hope this makes sense now. There's too much ink, so let me just kind of remove it. Okay. So we have already discussed lactate, we have discussed pyruvate. Now there are two molecules left, glycerol and alanine. So glycerol, as I said, it is from the breakdown of triacylglycerides, which are stored in the adipose tissue. So they will break down to give glycerols, a glycerol molecule and fatty acid molecules. Fatty acid molecules cannot be used as gluconeogenic precursors, because fatty acids on oxidation give you acetyl coenzyme A. They give acetyl coenzyme. What's wrong? They give acetyl coenzyme A. And acetyl coenzyme A is a 2-carbon molecule. This acetyl coenzyme A is fed into the TCA. But what happens during TCA? During TCA, you basically lose 2 CO2 molecules. So during TCA, you lose 2 CO2 molecules. And so that means that you are losing 2 carbon molecules. So if you use fatty acids as a precursor, then there is going to be no net reaction occurring, because you are feeding 2 carbons and you are losing 2 carbons. So there is actually nothing happening. You are not making those carbon molecules get utilized to form glucose molecule. So that is why fatty acids cannot be used as a precursor for gluconeogenesis, and that is also a reason why you cannot use acetyl coenzyme A, at least in the case of humans, as a precursor for gluconeogenesis. But at the same time, I said that propionate can be used as a precursor, and we are going to see how it is used as a precursor. But let's just focus on the glycerol molecule for now. So glycerol molecule enters the gluconeogenetic pathway in the form of dihydroxyacetone phosphate. So in the first step, it gets converted into glycerol 3-phosphate, and in the second step, it gets converted into DHAP, dihydroxyacetone phosphate. And by the way, this is just a detailed version. So you get glycerol to glycerol 3-phosphate. From glycerol 3-phosphate, you get DHAP, and DHAP can participate in the aldol condensation reaction, and it can thus be utilized to form glucose. And finally, we come to alanine. Alanine is a glucogenic amino acid. So alanine can get converted into pyruvate via transamination reaction. For this particular reaction, you are going to use alanine transaminase as the enzyme. It's a transamination reaction. And then the pyruvate is going to enter into the mitochondria. And then all the reactions are going to happen just like we discussed. So this is how you're going to use alanine. Apart from alanine, you also have other glucogenic amino acids, and all of them give one or the other intermediate of TCA. So, you can see all of these are actually giving pyruvate, so they can enter the the gluconeogenetic pathway as pyruvate. All of these are going to give you alpha ketoglutarate. So they're going to enter first the TCA, they're going to get converted into oxaloacetate, and then the other reactions are going to take place. And similarly, all of these amino acids, as you can see, they are converted into one or the other intermediate of TCA, and thus all of them can be used as a gluconeogenetic precursor. And as we have discussed, leucine and lysine, because they are strictly ketogenic, they will not be used and they cannot be used as a glucogenic precursor. And lastly, we come to propionate or propionyl coenzyme A rather. So propionyl coenzyme A is formed from the oxidation of fatty acids with an odd number of carbon atoms. So when you have fatty acids with even number of carbon atoms, then for example, say 16 carbon fatty acid, then when it gets completely oxidized, you're going to get 8 molecules of acetyl coenzyme A, because each acetyl coenzyme A molecule has 2 carbon atoms. But when, for example, you have 17 carbon atom fatty acid, that's an odd number of carbon atoms, you're going to get 8 molecules of acetyl coenzyme A, 7 molecules of acetyl coenzyme A, and then you're also going to get 1 molecule of propionyl coenzyme A, which contains 3 carbon atoms, right? Because 7 times 2 is 14, so you get 14 over here, and then 14 plus 3 is 17. So that's the maths. I just did the maths, but that's essentially where the propionyl coenzyme A is coming from. It's coming from the oxidation of fatty acids with odd number of carbon atoms, as well as it also comes from the oxidation of some amino acid. Now, when your propionyl coenzyme A is the molecule, it can eventually, by a series of reaction, get converted into succinyl coenzyme A. And so it can be fed into the Krebs cycle. But propionyl coenzyme A is not a major precursor in our case. It's a major precursor for gluconeogenesis in ruminants. And so it can be used as a gluconeogenetic precursor. Alright, so I hope that we have discussed all of the precursors. And this is just a diagram. I have already discussed with you that the reaction by glucose 6-phosphatase, the reaction catalyzed by glucose 6-phosphatase takes place in the ER. This is your enzyme complex actually. This is your protein complex. The T1 is a transporter which will transport glucose 6-phosphate into the lumen of the ER. Then this SP is actually a calcium binding protein which is required for stabilization of this glucose 6-phosphatase. And then this glucose 6-phosphatase is going to be the enzyme which actually catalyzes this reaction of conversion of glucose 6-phosphate into glucose. The inorganic phosphate which is released will leave the ER lumen through another transporter, and then the glucose is also going to leave the lumen via another transporter, and then it's going to eventually leave the cell to enter the bloodstream. And so thus they can regulate the blood glucose levels. The kidney cells and the liver cells can regulate the blood glucose levels. This completes our gluconeogenesis. I hope this video was helpful. I will see you guys in my next video.